This will give you the
Hi, my name is DevBot, also known as 3.3.
My name is Michaela Santos, also known as 3.20B.
Sophie Yang, 3.28.
3.14 Jennifer.
Alright, so we'll first be talking about what DNA is.
P3G5.
P3G5. DNA is a large double helical molecule that's found in all organisms on Earth, and found in the nuclei of most eukaryotes. Now, we'll first discuss what it is. This is DNA. Typically, it's in a coil, which is, it's in a coil. But this is the straight part. It's composed of nucleotides, which include a nitrogenous base, a pentase sugar, and a phosphate group. These phosphate groups are bonded using phosphodiester bonds, while the nucleotides of complementary bases, for example, cytosine and guanine and adenine to thymine, are bonded together using hydrogen bonds. This allows the molecules to not only remain flexible, but also undergo several other replication processes.
So in DNA replication, this is the leading strand and this is the lagging strand. This is the leading strand because it goes from 3 to 5. So the anti-parallel strand goes from 5 to 3, which is the direction of the replication. And this is the lagging strand because it goes from 5' to 3'. So the anti-parallel strand has to go from 3' to 5', which is pretty much like against the replication direction. So the nucleotides have to be formed backwards, which kind of delays the replication process. And yeah, okay. So helicase comes in to untwist the double helix and the single single-stranded proteins come in to keep the strands from coiling back together into a double helix. And then RNA primase comes in to add a nucleotide, adds in a primer to trigger DNA polymerase 3, which adds the rest of the nucleotides. So
RNA doesn't contain the nitrogenous base of thymine. Instead, it's replaced with uracil
and then
in mRNA.
topoisomerase rotates the strand to prevent tension in the DNA strands. And because since this is the lagging strand and the nucleotides have to be built backwards, they're called Okazaki fragments since there's gaps in between the nucleotides, which is stitched together by DNA ligase at the end, and DNA polymerase 1 takes away the primase and the primer.
Oh my god, okay. Okay. So, in the next step we have transcription. So in transcription, the RNA polymerase first finds the promoter, which every cell has a different one. So the RNA polymerase finds the promoter, in this case is the TATA, and then so it builds the nucleotides in the 5' to 3' direction. So it builds them. Okay. And then so once it goes to the end, to polyadenylation, the 5' cap is added, and then that's before, never mind. That comes after. We have termination first. Termination has to first occur. So in termination, there's like a hairpin right here, and then the rest of this like just falls off. The rest of the RNA just falls off. And then we have the post-transcriptional processing, in which the 5 cap is added, the polyadenylation, and then we have a poly-A tail that's added. So, and we have splicing.
Actually, Jennifer, if I can interject.
Yes.
The introns are cut out first, and then the cap and the tail.
Sorry. I apologize. I don't know where to cut either.
You labeled it over.
Sorry, guys. So this one is an intron, so this one falls off, and then the exons are now bounded together by spliceosomes, and then poly-A tail and the 5 cap are added, forming mature mRNA.
Okay. Now that we have mature mRNA, this binds to the small ribosomal complex. Okay. So the mRNA first goes to the cytoplasm, where it reaches. It exits the nucleus and then goes into the cytoplasm, where it reaches a small ribosomal. It binds to a small ribosomal unit and a large ribosomal unit. At the P site, a tRNA initiator tRNA molecule binds to the start codon of the mRNA. Okay. So first the tRNA molecule comes in, and then the large ribosomal subunit binds. So the initiator RNA molecule comes in with an amino acid at the start codon, and then at the P site. So, oh my gosh, sorry. As the ribosomal unit shifts down the mRNA, we add another amino acid along with another tRNA molecule along with an amino acid to the A site here. Then we shift down again by one more codon.
No, we have to.
Okay.
The peptide bond. The
peptide bond forms between the two amino acids, and then this first amino acid reaches the E site.
The amino acid is transferred to the
second amino acid, and then the first one goes to the E site where it's released. Then the second amino acid, along with the second tRNA molecule, along with the polypeptide chain that formed between the two amino acids, reaches the P site. And shifts downwards. Okay. We have another tRNA molecule with another amino acid. Then the polypeptide bond forms between these. This one it shifts. Sorry.
Okay. No. After the peptide bond, the amino acid that's formed, the polypeptide is shifted to the tRNA on the A side.
Mm-hmm. And then we shift. And then
the tRNA moves out.
Then it moves out the E site. And then the process continues down the chain. So,
actually, this is a.
The strand.
Yeah. This way.
Yeah.
Okay. Then go down again, and then the bond forms here, and then this one's released here until we reach. It should be here, and then the fourth one here.
So this is highly beneficial or not beneficial to any organism because it could either be a positive impact based on this change or a negative impact. As such, because sometimes a stop codon could be
until we reach the stop codon, which a release factor binds to with the. Oh yeah. Yeah. Oh yeah. Here's the release factor. And then the polypeptide chain, yeah, connects, and then the release factor binds at the A site, and then where's the
So let's say that the gene is changed to C, but because they're different are coded,
And then the polypeptide chain is
yeah,
released.
and then the whole thing dissociates.
Okay, and then we have
mutation. You haven't thrown in it yet, Sophie. Come on. The fun part's just getting started. Here. All right. Oh, you should probably come this way so it makes more sense.
sorry.
Alright, so here is just our example DNA strand, and here's our start codon, AUG, and our stop codon, UGA. And what occasionally happens to DNA or RNA strands is that they become mutated. That means somehow, some way, nucleotides come and interfere with the normal structure. Let's say this is the normal structure, okay? And one of the main types of mutations is called a substitution. So here, instead of having, let's say for example, a cytosine-guanine pair, it's substituted for a uracil-adenine pair, or a thiamine-adenine pair. It depends where it's happening. So this can sometimes cause issues where the start or stop codon is changed. So let's say, for example, we move this out of the way here. Now that means the enzyme reading the strand doesn't know where to start or where to stop. This can cause unnecessary elongation. This can cause the protein to not be coded at all if it's missing start codon. And this can generally cause a very big issue within the organism, or in rare cases, a very big benefit. Let's go back to the original structure. Now the other type is called a point mutation. This means that at a certain point, an insertion deletion mutation. This means that at a certain point, a nucleotide pair, a single nucleotide pair can be either inserted in, taken out, or it can happen in multiple groups. For example, let's say we add in this UA pair here. So what does this do? Well, now it has changed the entire reading frame. Originally, the enzyme reads it in a codon like this. Let's just remove this. 3, 3, 3. All it should be. But now once you've added this, you've added in an extra codon. Now the enzyme reads as 3, 3, 3, and so on and so forth. What happens is it ends up missing out on the stop codon. Now this can stop the protein from being created, or from being stopped at a right time, causing unnecessary elongation. Now, going back to substitution, if in the event that a substitution mutation does occur, let's put this back there. Now, in the event of a substitution mutation, mutations that are somewhat harmless, this means that according to all the amino acids that are being read during translation, for example, that the same amino acid is being translated regardless of the structure. That's called a missense. That's called a silent mutation. While where the wrong amino acid is created is called a missense mutation. In the events where the start or stop codon is substituted or changed, this is called a nonsense mutation. Now going back to the insertion/deletion, almost all mutations are harmful. However, because they shift the reading frame. However, if they occur at the end of a strand compared to the beginning of a strand, the harm is considerably less. Additionally, if you add in, for example, 3 more nucleotide pairs or remove 3 nucleotide pairs, the mutation essentially becomes a missense mutation unless the start and stop codons are affected. That way, the wrong amino acid is coded for, but it does not prohibit the protein from functioning normally, albeit it will be somewhat inefficient. So that's it for DNA mutations. Would any of you guys have anything else to add? No? Alright. Well, thank you.